will discuss the histology of the pancreas. At the end of the lecture, the student should be able to discuss the parts of the pancreas, discuss the features of the exocrine and endocrine pancreas. Now, firstly, what is exactly the pancreas? This is an organ, a retroperitoneal organ, extending from the concavity of the duodenum. So, the concavity of the duodenum on the right side of the spleen and on the left in the posterior abdominal wall retroperitoneally. The pancreas is covered by a connective tissue that is forming a capsule for it. The septa arising in the capsule extends into the gland, dividing it into many lobules. Now, this gland is partly exocrine and partly endocrine. The main bulk of the gland being constituted by its exocrine part. Exocrine part means that it contains the ductal system. The absence of ductal system is seen in the endocrine glands where these secretions are poured directly into the blood. The exocrine pancreas is secreting very important enzymes playing important role in the digestion of carbohydrates, proteins and fats, and after which these products are carried to the liver through the portal vein. If we see the sectional view of the pancreas, right, we can see it has both endocrine and the exocrine parts. The exocrine component forms majority of the pancreas and basically consisting of closely packed serous acini with zymogen cells arranged into small lobules, okay, like this over here. The lobules are surrounded by a thin intralobular septum. This is your interlobular septum, connective tissue septum, that is basically containing the blood vessels, the interlobular duct, the nerves, sensor receptor called the Paxinian receptor, Paxinian corpuscle. Now, when the serous acini, within the serous acini are isolated pancreatic islets of Langerhans. These pancreatic islets represent the endocrine portion and is the characteristic feature of the pancreas. Each pancreatic acinus, right, consists of a pyramidal shaped protein secreting zymogenic cell that is surrounding a central lumen. The excretory ducts of the individual acini are visible as pale staining centroacinar cells within the lumen. The secretory products leave the acinus via the intercalated or the intralobular duct that have a small lumina lined with low cuboidal epithelium, right? So I'd like to show you this. Yeah, so can you see this? This is one particular serous acini, right? We have the zymogen cells. Can you see their pyramidal zymogen cells at the lumen? And then the intercalated duct is continuous with the centroacinar cells over here. Okay. So you could see in the previous picture the pancreatic acinus containing the pyramidal shaped protein secreting zymogenic cells surrounding a small central lumen. Right, and you saw the centroacinar, pale staining centroacinar cells within the lumen itself. Right. So now the intercalated ducts drain into the interlobular duct. Here you can see centroacinar cells in the pale staining ones in the center. Right. This is a Paxinian corpuscle. Right. Now the interlobular ducts are lined by simple cuboidal epithelium and become taller and stratified in larger ducts. The pancreatic islets are demarcated from the surrounding exocrine acinous tissue by thin layer of reticular fibers. The islets are larger than the acini as clearly visible over here and are compact clusters of epithelial cells permeated by capillaries. You can see these red things over here, they are the capillaries, and they have numerous capillaries simply because they are endocrine in function. They are draining directly into the bloodstream. Right? This is a sectional view of the endocrine and the exocrine pancreas in low magnification, hematoxylin and eosin stain. So now coming to the pancreatic islet, right? So it is pale staining as you can see, pancreatic islet. It is illustrated in high magnification over here. The endocrine cells of the islets are arranged in cords or clumps, between which are found the connective tissue fibers and the capillary network. Right? You can see the capillary network. These are the connective tissue capsule formed by reticular cells. This is the intercalated duct cells of the pancreatic islet. These are the capillaries. This is the connective tissue capsule, and this is the centroacinar cells in the serous acini. Now, you can see a thin connective tissue capsule separating the exocrine pancreas from the endocrine pancreas. Some serous acini contain a pale staining centroacinar, as the name suggests, center of an acinus, which are part of the duct system that connected to the intercalated duct. Right. Myoepithelial cells do not surround these secretory acini in the pancreas. In routine histological preparations, the hormone individual cells secreting of pancreas cannot be identified. Okay, right. So again, let's see. This is the intercalated duct cells of the pancreatic islets, but the individual cells secreting hormones is not clearly defined in H&E stain. We have the capillary network. This is the connective tissue capsule, and this is your centroacinar cells in the serous acini. Okay, so because we cannot identify the individual cells in H&E stain, this pancreas has been prepared by a special stain to distinguish the glucagon secreting alpha cells from the insulin secreting beta cells, right? Capillaries, yes, simply because it is an endocrine organ. Now, the cytoplasm of the alpha cells stains pink, whereas the cytoplasm of the beta cells is staining blue. The alpha cells are situated peripherally. Here they are situated peripherally in the islet, and the beta cells are more in the center. Also, the beta cells predominate, constituting about 70% of the islet. We also have delta cells which are not illustrated in this picture. Okay, they are the least abundant. They have a variable cell shape and may be found anywhere in the pancreatic islet. Now, as you can see, there are numerous capillaries demonstrating the rich vascularity of the pancreatic islet. We have a thin connective tissue capsule separating the islet from the serous acini, and the centroacinar cells are visible in some of the acini over here. Right. So the special preparation is called Gomori's chrome, alum hematoxin, and phylloxene stain for staining of the pancreatic islet. Now, the endocrine component of the pancreas are scattered throughout the organ as islands of endocrine cells called pancreatic islets. They are secreting two major hormones that regulate the blood glucose level and glucose metabolism. The alpha cells in the pancreatic islet produce the hormone glucagon, which is releasing in response to low level of glucose in the blood. Whereas also glucagon elevates blood glucose level by accelerating the conversion of glycogen, amino acid, and fatty acids in the liver cells into glucose. Whereas the beta cells in the pancreatic islet produce the hormone very important insulin, which is released when stimulated by elevated blood glucose level after a meal. Okay, so again have a look at the alpha cells at the periphery secreting glucagon and the beta cells in the center secreting insulin. Now, what is the function of insulin? It is lowering the blood glucose levels by accelerating membrane transport of glucose into liver cells, muscle cells, and the adipose cells. Insulin also accelerates the conversion of glucose into glycogen in liver cells. The effect of insulin on blood glucose level are obviously opposite to that of the glucagon. We also spoke about the delta cells. Its function is to secrete the hormone somatostatin. This somatostatin decreases and inhibits the secretory activity of both alpha and beta through the local action within the pancreatic islet. We also have the pancreatic polypeptide cells producing the hormone pancreatic polypeptide, which inhibits production of pancreatic enzymes and alkaline secretions. Okay, so you can see this is a periodic acid shaped stain, right? You can see the pancreatic islet over here, right? The intercalated duct, connective tissue capsule surrounding the pancreatic islet, right? Centroacinar cells in the middle, and then we have the secretory acini, and of course, numerous capillary network. Right, this is a simpler form of pancreas. You can see, right? This picture you've seen before. Now, just to revise, this pancreas lies in the duodenal loop. The exocrine components forms majority of the organ composed of serous acini. The zymogen cells of acini filled with granules that contain the digestive enzymes. The acini containing pale staining centroacinar cells in the lumina. The centroacinar cells are continuous with the cells of the intercalated duct. Hormones secretin and cholecystokinin are regulating its secretions. The intestinal enteroendocrine cells release hormones when acid chyme is present. Now, secretin stimulates sodium bicarbonate production by the centroacinar cells and the intercalated duct cells. Alkaline sodium bicarbonate fluid neutralizes the acidic chyme. The cholecystokinin released when fats and proteins are present in chyme. Also, cholecystokinin stimulates the production of the pancreatic digestive enzymes, and these enzymes are produced firstly in an inactive form and gets activated in the duodenum. Coming to the endocrine part of the pancreas, it is form of isolated pancreatic islet surrounded by fine reticular fibers. There are 4 types of cells present over here: the alpha, beta, delta, and the PP cells. Alpha produces glucagon in response to low sugar levels. Glucagon elevates blood glucose by accelerating conversion of glycogen in liver. The beta cells produce insulin during elevated glucose levels. Insulin lowers the blood glucose by inducing glucose transport into the liver, muscle, and adipose cells. The delta cells produce somatostatin, which inhibits activity of both alpha and beta. And the PP, or the pancreatic polypeptide cells, inhibit enzymatic and alkaline pancreatic secretion. So that's all about the histology of the pancreas. Thank you.